So here's an explanation of protein synthesis from start to finish. So let's look in the upper left-hand corner. We're going to take a look at an animal cell. I can tell it's an animal cell because of the shape and the fact that there is no cell wall. So we're going to zoom in on the nucleus. So I like how this picture kind of zoomed in. And the nucleus contains the DNA, which is the ultimate instructions for the activities of the cell. So it contains the instructions that tells what the cell is going to make, how it's going to make it, when it's going to make it. So we're going to be focusing on the production of ribosomes themselves. We know that ribosomes will ultimately make proteins, but this is where the ribosomes themselves will be made. So if we zoom in on the very center of the nucleus, you're going to see how the nucleolus is kind of the dense center of the nucleus, and this is where the ribosomes, another name would be rRNA, is actually made. Okay, so we have a ribosome that's made in the nucleolus. Now it needs to travel out of the nucleus, either into the cytoplasm or onto the rough ER. So if you'll notice that in the nuclear envelope, which is the outer portion of the nucleus, here, there are these tiny little holes. These are called nuclear pores. So our ribosome that we made is now going to exit out of these little holes, and it's going to travel into the cytoplasm. Or it could then exit into one of those holes, actually flow into the rough ER. So now let's look at the larger picture down here. And so same thing. So you've got the nucleolus in the center, that's where the ribosome is made. It will then travel either out of the nucleopore into the cytoplasm, or it travels into the rough endoplasmic reticulum. And you'll see we have all of these little dots here. Those represent all the different ribosomes. Okay, it's studied on the ER. Okay, so there, all those little dots, all those ribosomes. Okay, so there's our ribosomes. That's where the protein will be made. So the proteins are made there, and say it's a protein that needs to be transported out of the cell. That protein that we make at the ribosome will then travel. You see these are like little vesicles. These are little circles here. These are membrane bound. So it's a very thin layer, just like the cell membranes layer. It's a lipid bilayer. And it will come into the Golgi body. And the Golgi body, I know in picture they call it the Golgi complex. They are going to package this protein. So they're going to put a little a membrane layer around it called a vesicle, and they'll put labels on it, ID tags, so that it knows where to travel and where to travel, how to travel. And then it will bud out of the Golgi when it's ready, and it will exit out of the cell through exocytosis. Exo meaning exit. And that is how the protein basically starts from the inside of the nucleolus. The ribosome is made in the nucleolus. The ribosome travels to the outside of the nucleus to the rough ER, or the cytoplasm. There protein is made. It can then go to the Golgi apparatus where it gets packaged, and then the Golgi apparatus will help export it or transport it out of the cell.